Optical tweezers are scientific instruments that make use of focused laser beams to provide an attractive and repulsive force, relying on the reflective index of the object to trap, manipulate and position micron-sized objects. The idea of using optical tweezers as a method for trapping atoms was first discovered by Arthur Ashkin in 1970 and were originally known as single-beam gradient force traps. Today, optical tweezers have a wide range of applications in the area of biological sciences, allowing the study of a variety of biological systems. But how do optical tweezers work? The basic principle behind the tweezers is the following. Lasers are focused through a series of lenses, where the laser light applies a force on the particles in the beam along the direction of beam propagation. Refraction occurs as the laser light enters and exits the dielectric bead. As a result, the light and the momentum it is associated with changes direction as it exits the bead. Due to Newton's third law, there is an equal and opposite momentum acting on the particle which traps the bead in place. When the particle is near the center of lens, the net force on the sphere is pointed towards the center of lens. Thus, the trapping happens. One thing to note is that this could only be done when the dielectric beads are dissolved in a solvent. Moreover, the refractive index, n, of the particle should be bigger than the solvent's 1. This explains why it is difficult to trap metallic microspheres, which refractive index is small. Also, if there is an air bubble in the liquid, the resulting net force points downwards, because the refractive index of air is less than the reflective index of water. Optical tweezers setup contains a laser, a beam expander, mirrors, a microscope objective and a camera. However, these are very complicated instruments that require a working knowledge of microscopy, optics, and laser techniques. In the experiment, 532 nanometers laser was used. 1, 3 and 5 microns sized particles were examined. The particle's size was bigger than the laser's wavelength. Therefore, the large size limit regime was applied in the experiment. It is possible to determine the trapping force due to the viscous drag of optical tweezers, using the equation F equals to 6 pi nRV, where R is the radius of the microsphere, N is the viscosity of water, and V is the terminal velocity. All the variables are kept constant as the microspheres are dissolved in water. We just have to measure the terminal velocity and we will know the viscous drag. The terminal velocity here is the velocity at which the microsphere becomes dislodged from the optical tweezers. By varying the speed at which the optical tweezers moves across a specified distance and monitoring when the microsphere leaves the influence of our optical tweezers, we are able to find its terminal velocity. Note that the influence of nearby walls in the sample set is ignored. In the experiment, 1 micron-sized trapped particle is shown, moving with the velocity of 12 micron per second. As it is seen, the particle stays trapped. Now the same particle is moving with the velocity of 15 micron per second and becomes dislodged. Thus, the force of the 1 micron-sized particle is 0.28 piconewtons. The experiment is repeated for the 3 micron-sized particle. The particle stays trapped at the velocity of 30 micron per second and becomes dislodged at the velocity of 40 micron per second. The force was found to be 2.27 piconewtons. The trapping force of the optical tweezers was also found using the 5 micron sized particle, which became dislodged at the velocity of 42 micron per second. And the force was 3.97 piconewtons. The experimental results agree with the theoretical ones. The trapping force becomes larger as the radius of a particle increases, since the radius is directly proportional to the trapping force. Moreover, the experiment was repeated for 2 particles. The lower escape velocity of 31 micron per second was recorded. For 3 particles, even lower escape velocity of 20 micron per second was found. It was concluded that with more particles, the intensity of the laser would be more spread out, and thus, not having enough power to effectively trap multiple particles as compared to trapping a single particle. In the next experiment, we tried to manipulate the particles to form a letter A by finding a loose particle, picking it up and shifting it to the desired location and releasing it. we are able to pick up and release the particles by simply blocking the light source with a makeshift board. The experiment challenging, as firstly, there were dead particles, which could not be picked up. This was due to the particle being stuck on the surface of the slide and was not free to move within the sample. Secondly, with the particle more free to move about, we are unable to control its free movement after releasing them, causing a distorted instead of the intended one. In summary, optical tweezers are scientific instruments that use a highly focused laser beam to provide an attractive or repulsive force. Optical tweezers are used to trap dielectric spheres, viruses, bacteria, living cells, organelles, and even strands of DNA. Applications include organization and tracking of movement, for example, of bacteria, measurement of small forces, and altering of larger structures such as cell membranes. The physical properties of DNA can be examined via optical tweezers by attaching the beads to the ends of single pieces of DNA. Experiments have measured the elasticity of the DNA, as well as the forces under which the DNA breaks or undergoes a phase transition. In the experiment, we have shown how micron-sized particles can be manipulated via optical tweezers. Thank you for watching.